In this session, our guest speaker will be discussing his presentation titled Whole Exome Sequencing Reveals Timing and Origins of Local and Distant Metastasis in Lung Cancer. Dr. Bo is the Director of R&D at Geneseeq Technology and is responsible for the development of NGS testing products, computational methods, and mathematical modeling in cancer genomics. He holds more than 10 patents and has published numerous papers in peer-reviewed journals including Gut, Journal of Thoracic Oncology, Clinical Cancer Research, Oncogene, and Bioinformatics. He is also an active member of ASCO and AACR. I'd like to thank Dr. Bo for his time in putting together today's presentation. We will now begin today's session.
My name is Hua Bo. I am the Director of R&D from Geneseeq Technology Inc. It's my pleasure to give the presentation of this collaboration webinar between Integrated DNA Technology and Geneseeq. My topic today is whole exome sequencing reveals timing and origins of local and distant metastasis in lung cancer. I'd like to first give some background of primary lung cancer. Lung cancer is the leading cause of cancer-related deaths in the world. In the United States, it's estimated that 250,000 patients were diagnosed with lung cancer, with 150,000 deaths due to lung cancer per year. So the treatment for lung cancer mainly includes surgical resection, chemotherapy, target therapy, and immunotherapy. Large-scale sequencing studies have revealed complex genomic landscape of lung cancer. For example, mutations in EGFR, KRAS, and ALK fusions are mutually exclusive in non-adenocarcinoma patients. And it's well known that Asian patients have higher proportion of EGFR mutations, and the Caucasian population have more KRAS mutations. The deeper understanding of the genetics of lung cancer has led to the development of small molecules that target these somatic mutations, and these targeted therapies have significantly improved the overall survival of lung cancer patients. So most lung cancer patients were diagnosed at stage 4, and it means these patients already developed metastasis. Most common sites are brain and bone metastasis. Although gene alterations have been established driven primary non-tumor formation like EGFR KRAS shown in the previous slide, it's not well known what are driver alterations contribute to the development of metastasis. So discovery of these metastatic associated drivers can help us diagnose this much earlier and provide more precise treatment. Further, it's not known whether disseminated cancer cells seed metastasis before primary non-tumor is clinically detectable or not. So this question is related to when metastasis first occur. Determining the metastatic timing is important to schedule the cancer screening and the direct surgical treatment to minimize the incidence of metastasis. And finally, where distant metastases come from? So the traditional model in oncology is that lymph node metastases give rise to distant metastases, but some contradicting results were observed in colorectal cancers. And more recently, a number of clinical trials have shown that lymph node removal does not always improve patient survival. These findings inspired the alternate view that lymph node metastases do not give rise to distant metastases and suggest that treatment strategies may need to be reevaluated. So in this study, we would like to answer 3 questions. The first, identifying metastatic driver genes. And the second, using mathematical model to estimate the primary tumor size when metastatic seeding first occur. And the third one, analysis of evolutionary origins of distant metastasis, whether it comes from lymph node metastasis or directly from the primary tumor. So to answer these questions, we recruited 47 Asian lung cancer patients and 40 patients with paired primary and metastatic tumors, and 7 patients with only metastatic tumors. Metastasis sites include brain, pleural, lymph node, adrenal gland, and bone. If possible, we perform multi-region sequencing for each tumor to better characterize subclonal mutations. So to better answer these questions, we rely on estimation of cancer cell fraction of coding somatic mutations and copy number alterations in tumor samples. And whole exome NGS is the best option for this purpose. First, using NGS technology gives us more comprehensive data and more discovery power, and then can be achieved through PCR of limited number of genes. And whole exome sequence, typically 150 coverage depths, provides more power to identify low-frequency functional subclonal mutations compared to whole genome sequence, typically only 20 coverage depths. WES sequence pipeline starts with genomic DNA extraction and library preparation. We use IDT xGen Exome Research Panel V1 to enrich exonic sequences, and this panel spans a 39 megabase targeted region, about 20,000 genes of the human genome. And each probe within the exome panel was individually synthesized and underwent a strict manufacturing and quality assessment process in accordance with GMP standards. By using this optimized protocol, we can obtain high quality, reliable, and uniform exome data. We sequenced 150 depth coverage for tumor sample and 60 depth coverage for blood control samples using Illumina HiSeq 4000 platform. So detection of somatic mutations is a key step in characterizing the cancer genome. We used MUTAC tool from Broad MIT Institute for identifying single nucleotide variants and short indels with different variant allele frequencies exome sequence data. Paired normal blood samples was used for filtering germline variations, and a panel of normal samples were used for filtering recurrent common artifacts of false positive mutations. And a new specific code number analysis from exome data can greatly extend the utility of NGS beyond the identification of mutations for the detection of homozygous, heterozygous deletions, copy neutral loss of heterozygosity, and new specific amplifications. So here we use the FACES package from MSK Cancer Center to identify somatic copy number alterations in tumor samples using paired normal blood samples control. So this OncoPlot plot shows the clonal and subclonal status of gene mutations, gene copy number changes, and arm level copy number changes in our cohort. As we can see, the most frequent mutant genes in our non-cancer cohort were TP53, EGFR, KRAS, PIK3CA, and KEAP1, which mainly contain clonal alterations. And these gene mutations were generally identified in both the primary and the metastatic tumors within a patient. You can also see chromosome arm level gain and loss are common in primary and metastatic tumors. Again, I need to mention high quality uniform sequence data from WES are very important for getting these accurate somatic alterations in the tumor samples. The overall concordance between primary and metastatic tumor mutations was 45%, ranging from 7% to 81% per patient. Lymph node metastasis showed the lowest proportion of shared alterations. In other words, highest divergence, while distant metastases have higher proportion of shared alterations. It suggests lymph node metastases may diverge from primary more earlier compared to distant metastases. And here is some other genomic patterns and metrics among primary and different metastatic tumor samples. In general, distant metastases have higher ploidy and more unstable genomes compared to primary tumors. And some metastatic site-specific differences were also observed, such as plural metastasis has lowest tumor mutation burden. Green metastases have highest chromosome instability and ploidy. In order to answer the first question of identifying somatic events driving non-cancer metastasis, we define the metastatic selection landscape of each event in each patient. So there are three types of events: metastatic selected, maintained, and unselected. If a somatic event in clones showing increased frequency in metastatic tumor compared to primary tumor, then it's under positive selection associated with metastasis. These metastatic selection events include alterations that are subclonal or clonal in metastatic tumors but absent in the primary tumor. And those are subclonal in primary tumor but becomes clonal in metastatic tumor. From the population dataset, we can test whether some of these events are statistically enriched in the selected group compared to non-selected group. We identify several gene copy number alterations like Richter NKX2 and MYC amplification associated with non-cancer metastasis. In addition to gene level copy number alteration, chromosome arm level copy number changes also contribute to metastasis, such as 6p and 11p loss and 20p gain. These results suggest non-cancer metastases were mainly driven by copy number changes. Interestingly, MYC and Richter have target drugs and may be potential therapeutic targets. Among the metastatic-driven alterations, we further identify MYC amplification was significantly associated with worse overall survival in our cohort and MSK cohort from online dataset. MYC amplification might have broader effects on non-cancer metastases, promoting the migration to multiple different organs. But besides non-cancer, MYC amplification has been reported to promote metastasis in many other cancers, including breast cancer, colorectal cancer, which could be partially owing to its regulation of EMT transition genes. Now we move to the second question we'd like to answer. So, estimation of the primary tumor size when metastatic seeding first occur. Here we use the spatial computational model of tumor progression and the Bayesian statistical inference framework to time dissemination in a patient-specific manner. We simulate the tumor cell growth, dissemination, and the lineage relationships of tumors under different parameters. And we can decide which parameter combinations best explain the observed mutation patterns. Because the tumor volume of 1 cubic centimeter, approximately 100 million cells, is usually considered as clinically detectable. So the estimate ND here, cell number of primary tumor when metastasis first occur. So this value could be used as an indicator for the dissemination time, with cell number less than 100 million indicating early dissemination. So it means metastasis happened before the primary tumor became detectable. And when cell number greater than 100 million, indicating late dissemination. And the heat map on the right represents the estimated cell numbers when metastasis first occur for each patient. And as you can see, lymph node metastasis account for a large fraction of early dissemination in our cohort. And the size of their matched primary tumors at initial dissemination was relatively small, considered as clinically undetectable. But plural metastases show equal proportion of early and late dissemination, and more distant metastases like bone brain show the trend of higher proportion of late dissemination when the primary tumor is clinically detectable. In terms of years, lymph node metastatic seeding happened approximately 4 years before clinical diagnosis, and distant metastasis seeding happened 2 years before diagnosis. The one explanation of this difference between lymph node and distant metastasis is establishing lymph node metastasis may be a more efficient process compared to establishing distant metastasis, and may therefore happen earlier and more frequently. And this result suggests that it's possible to prevent non-cancer metastasis by early cancer detection. The current NCCN guideline recommends annual low-dose CT screening for people with high risk of lung cancer. Given that some novel molecular technologies such as circular cell-free DNA method are potentially more sensitive than low-dose CT to detect cancer, adopting this sensitive cancer diagnostic method could be important to prevent non-cancer metastasis in the risk population. And the last question we investigate is whether distant metastases are seeded by lymph node or directly seeded by the primary tumor. And in our cohort, we have 8 patients have both lymph node and distant metastasis. This gives us a good opportunity to answer this question. So using somatic mutation data, we built phylogenetic trees and examined the evolutionary relationship between primary tumor lymph node and the distant metastasis for each patient. If the distant metastasis and the lymph node metastasis is within the same clade on the tree, it suggests the lymph node metastasis gave rise to distant metastasis. So it's common origin. Otherwise, lymph node and distant metastasis were each more closely related to a primary tumor region than to each other. So this is distinct origin. And interestingly, observed 7 out of 8 patients with distinct independent origins of lymph node and distant metastasis. And this result suggests that most cells in primary tumor have the ability to metastasize. In such tumors, all cells that disseminate would have an increased likelihood of colonizing distant sites. So in the future, it's important to determine whether cancers in the common and distinct origin categories exhibit different clinical behaviors. Also, if lymph node metastases do not always give rise to distant metastases, treatment strategies may need to be re-evaluated also. So the conclusions from this study: whole exome sequence provides an efficient and reliable approach for cancer genomic evolution studies. We identify several driver events contribute to non-cancer metastasis. Meek and Richter may be potential therapeutic targets, and distant metastases tend to have late metastatic seeding when the primary non-tumor is clinically detectable. So it's possible to prevent non-cancer metastasis by early cancer detection. And distant metastases were mainly seeded by primary non-tumor rather than the earlier colonized lymph node metastases. However, this result need to be confirmed by another patient cohort with multiple metastasis sites. And that's all for my presentation. Thank you so much for interest and attention.